estimated time for a molecule of glucose to move from the blood vessel, say, to a neuron is something like 2 seconds. So diffusion distances are minimized. We now know quite accurately how blood-brain barrier is formed. Here's a diagrammatic representation, cross-section of brain microvessel. Here's epithelial cell forming the microvessel. Here are astrocytic end feet, which are very closely applied to these blood vessels. Here's a small cell, a pericyte, which are quite commonly seen applied to the outside of the epithelial cells. And the blood-brain barrier is essentially formed by formation of tight junctions here.
Do you think this was a summary of your
Yes, exactly. I feel very much honored that the thesis he said that that was in accordance with the thesis, which is great.
okay? So
Really
it's
nice.
section up.
Start some formal questions. It might be useful, Hari, if you've got your thesis on the lab, because I'm going to refer to specific pages in the thesis on which my questions are based. And for those of you in the audience who have a copy of the thesis, this might also be useful. So my first question refers to page 9 of the thesis, which is your introductory section. And you mention here, I think very interestingly, that heat stroke or heat stress is actually the 3rd largest killer in the world as far as CNS causes are concerned, after traumatic and cardiovascular insults to the brain. And I just wonder if you'd like to expand on this a little more, because I think a number of us don't fully appreciate quite how common heat stress is, especially those of us who live in rather temperate climates. So I think it's a very interesting statement. I wonder if you'd like to say a few more words about that.
Well, I can only say in this case that more than half of the world population is exposed to the warm temperature, and usually the temperature ranges during summer. It is going to be from minimum in summer season 30 and then plus. For instance, 41 and in some cases 45 in Spain. We have been last week, and the temperature one can read was 42 degrees Celsius on the street. And therefore, the problem is that many persons, particularly, and now we know that we have a large population or significant population of having arterial disease or having some other hormonal disease, particularly like diabetes and hypertension. And these persons are much more prone to hyperthermic injury. They have much more significant effect to the heat stress. And therefore, and also there are many young athletes doing their exercise, routine exercise in the United States and different parts in the United Kingdom, for instance. New military recruits, they are very doing their normal exercise. And we have evidences published in Lancet and many other good journals that they die immediately when they exercise. And they are otherwise physically fit. So in these conditions, they are physically fit, they cannot cope with the stress, either the football players or military recruits or many other athletes. And therefore these numbers are really very high. So some normal healthy animals, followed by persons having different kind of chronic diseases. So all together, when they are exposed to this kind of environment, they die due to complications of their own disease, number one, or suddenly hyperthermia, which cannot be controlled, because until now we don't have any evidence or any drug that can reduce or help them. So the point is that, therefore, this is a statistics from American published database I collected, and according to them it is said that after cardiovascular, sudden heart infarct or that death, and then brain and spinal cord and brain injuries like motor vehicle accidents or fall, heat stress induced hyperthermic death is the 3rd largest killer that involves CNS.
Thank you very much. I think also it makes you've made the point, I think, very this relates to people who survive a heat stress. And you mentioned here that they have persistent mental disabilities. They have persistent brain damage. And I wondered whether this is sufficiently similar to be a consistent syndrome. In other words, can a particular form of mental disability be related to someone who has suffered heat stress, or is it very variable from person to person? So is this mental damage non-specific, or is there a specific syndrome which can be recognized?
Well, we have some sporadic case reports, and it suggests that this mental abnormalities is not a generalized phenomenon. We cannot say that if we are going to have some mental symptom, this is particularly sensitive to hyperthermic stress. But probably, I mean, it depends on what is people suffer. I mean, if a person gets heat stroke or hyperthermia at childhood, then probably, I mean, the intellectual functions are deteriorated. And we have evidence, for instance, that hippocampus is damaged, and in some cases, I mean, it is very well associated with epilepsy. And then this is another form of damage. So it is not universal, it is not similar, and it depends on the severity and magnitude of the hyperthermic insult at the beginning that can really predict the damage. So it can vary depending on individual and of course their previous healthy or disease states.
So there's no particular syndrome?
Not to our knowledge at the moment.
Thank you. The other thing that caught my eye was on page 15. We've got table 3 here, which shows the critical air temperatures and the body temperatures for a number of species, including man. And my question here was the question of the thermoneutral zone for man. And this is apparently quite wide. This surprised me. It's 24 to 31. And I think a number of sources claim the thermoneutral zone for man is much, much more narrow than that. Are these figures for a clothed man or a naked man? Because I think this does alter the size of the thermoneutral zone. Really quite significant.
Yes, and I think it's a very important question that you have raised here. And thermoneutral zone, of course, we have seen, and there is no any agreement still by the Thermal Physiology Commission. So they are still debating that can we put an ideal or thermoneutral zone for human beings, because we know that the thermal perception is different, and there has been so many studies that, for instance, the thermal sensation of, for instance, I should say that people living in Britain, for instance, and people living in Sweden. It has been shown, I think, 35 years ago. There is a book, Man in Hot Environment, and they have meticulously shown that people living in Sweden, they have very less thermal induced, I mean, they are more sensitive to any thermal load. So for them, I mean, if 26 to 29 could be obvious, they can feel stressed. But for instance, people have, from India, for instance, and South Africa, so they do not show changes in their basal metabolic rate. Even the ambient temperature is increasing to, for instance, 30 degrees Celsius. So there is a debate on Thermal Physiology Commission, and even the debate continued last week. We have this thermal regulation meeting in Seville. So what we have seen here, that we have plotted the minimum and the maximum, and that is the range from 24 to 31. And I believe that for average temperate climate, as you are familiar with that, for instance, 26 to 29 would be the ideal. But there are some other cases, like people living in much northern countries like Alaska, for instance, their basal metabolic rate can be enhanced even by exposing to 24. So this is a general variation, 24 to 31.
It's an interesting observation. I wonder whether you might like to just comment on this in general, but mammals generally seem to be able to adapt to lower ambient temperatures much more effectively and higher. You can only
our brain both cold sensitive neurons and warm sensitive neurons. And there is prevailing view that what happens in hyperthermia, that the warm sensitive neurons, what they do function, we do not know. But it is a common understanding at this stage that when we are going to have hyperthermia, the warm sensitive neurons are switched off. That's the one defense mechanism. And then when they do not feel warm sensation, then we have the mechanism of activated by cold, and then body try to lose heat. And therefore, our body temperature, for instance, is set on 37, and we have very narrow limit for killing. It's 5 degrees Celsius more. So I mean 42 degrees Celsius is much more effective. So that's why there is a common phenomenon that we can tolerate very much cold, because all our thermoregulatory mechanisms are mainly activated by the activation of cold sensitive. And when we have hyperthermia, warm sensory neurons are usually switched off. This is the convention at this moment. So therefore, probably we are much more sensitive to hyperthermia rather than the cold environment.
You now turn to page 22. Yes, it might be. That's it.
It'd be easier for everyone to read.
And on page 22, you're discussing the blood-brain barrier stressful conditions and the involvement of corticosteroids. Now, dexamethasone has been given for many years as a drug to treat cerebral edema in humans. And we know from various experimental studies that glucocorticoids tend to tighten rather than open the blood-brain barrier. And in fact, recent in vitro studies have shown that if you add glucocorticoids, such as hydrocortisone, to a monolayer or cerebral epithelial cells, they will form like junctions, and the transient epithelial resistance will increase quite significantly. Now, cortisol and other glucocorticoids are obviously involved with stress responses and released under stressful conditions. And therefore, this oppositional effect of glucocorticoids to blood-brain barrier opening are going to be part of the stress response. So they're going to tend to oppose initially the effects of heat stress. And I wonder whether one can think of, say, the blood-brain barrier polarization in heat stress as really part of how the cellular definition of the general adaptation syndrome, where one sees a state of exhaustion. So what's happening at 1 and 2 hours when initially stress reacts is that one has the protective stress response, which is the release of certainly the rat corticosterone, which is attacking the blood-brain barrier. And then this final open one is a phase of exhaustion. So would you like to comment on that? Is that, do you think, a good way of looking at this?
Yeah, well, this is a very important question, and I must admit that we have not been able to measure corticosterone in our heat stress rats in plasma. What is their level after 1, 2, or 4 hours of heat stress? But what we have some information on unpublished observation. We try to give animals a low dose of corticosterone at 4 hours, and this is 4 milligram per kg subcutaneously. That was quite similar. We calculated the level that is rising in after stress. We have no evidence how much the level rises in heat stress. So I can see that up to 1 and 2 hours of heat stress, we do not see permeability increase. Probably at that time, this level is very high. But at the end of 4 hours, probably it is very much decreased, and it could be overpowered by so many other destructive factors or chemicals, because we can also do not see rise in serotonin in 1 or 2 hours. So probably, I mean, this corticosterone is initially protecting, and when this effect is completely gone and the animals reach as the stage of exhaustion, then, I mean, they have no other way, and then the pathological response is opening of the blood-brain barrier permeability.
then possibly diffusing out across the damaged blood-brain barrier into blood. Now, you haven't mentioned S100 protein in the thesis, so I just wondered if anything is known about S100 in heat stress. What is happening to this protein?
This is a very relevant question concerning glial reaction in hyperthermia. And I can tell that according to our knowledge, even in the field of thermoregulation, people are not thinking the role of glial cells in thermoregulation. Though we know that glial cells are about 10 times more in number. And then regarding, so even GFAP in hyperthermia, people have not used, there are some studies in cell culture. So what we are looking at, we have seen GFAP increase at 4 hours, and then we wanted to study bimentin, and we have seen that it can be reoccurred after 4 hours of heat stress, showing damage. But we have also studied an unpublished observation about S100. We have thought similarly. But at the moment, this study is not complete. And we have seen changes in some regions, like hippocampus, it is increased, and in some cases in midbrain it is downregulated. So at the moment, I think we can see that in hyperthermia, S100 is definitely affected. But the pattern of its influence and different brain regions, we do not have any clear conclusion. So I think we need further study to do that before I can say anything about its role in S100 protein in hyperthermia. But still, it is influenced.
So there is an effect, certainly.
Yes.
Because it occurred to me, and I think this is something that might be worth considering, is that because we know in insult to the brain, traumatic injury, and cardiopulmonary bypass, the S100 increases in plasma. And this might actually be quite a useful indicator of thermal stress in the human.
That is really a good idea.
Because one could, by simply taking a blood sample, measuring S100, without any sort of invasive measurements of what's going on in a patient's brain, one might be able to get some measure of how severe the heat stress has been in an individual. So this might actually be quite an important area of future study.
Yeah, I think this we have never thought, but thank you very much. This could be used.
Because it would be a peripheral marker, which is used to be used.
Yeah, certainly. It's a very good idea. Surely we will look.
The next question just simply relates to the next page, where we're talking about heat shock protein and the response of heat shock protein. And I just wanted to ask you here really quite a specific question about where this heat shock protein may be coming from. Because I think in the thesis you talk very much about a possible upregulation of heat shock protein synthesis. But we know that within cells, heat shock protein and another protein called immunophilin, whose function I don't think is known, is complex to the intracellular cytoplasmic steroid receptor. So initially there's a triple complex of the steroid receptor, heat shock protein, and immunophilin. And when a steroid binds to the steroid receptor, immunophilin and heat shock protein dissociate. So what I am hypothesizing is that when you heat stress your animals, you have corticosteroid release by the adrenal, which enters the brain, it binds to the glucocorticoid receptor in the brain cells, and the heat shock protein associated with the corticosteroid receptor dissociates. Now when you see the increase in the heat shock protein, that may be as a result of this dissociation. In other words, when it's associated with the receptor, the epitope or antibody will be visible. You can't say.
Thank you for this proposition today. And most of us know that scrutinize Swedish thesis is very demanding. You have to master a field where the defendant have been working for years and
passed or not passed. But at that time it was graded.
Actually graded, yes.
So, and,
Yes, we've never done that in the British system. It's just the first degree is graded,
Uh-huh.
but the doctorates and
Mm,
they're just passed, they're not passed. I think one or two universities sometimes give cum laude, give honours. So if it's an exceptional thesis, it may get a special.
I see, yes,
special
yes,
mention.
yes, yes.
But I think that's unusual. Only 2 or 3 universities do this.
That time, if you had a good dissertation at above a certain of these remarks, then it would be easier at that time if you would apply for what they call the assistant professor or what they had docent in Sweden.